wanna tell.
Molecular cloning of drug targets. Hey everyone! Welcome back to our channel. Today, we're diving into an exciting topic in the world of molecular pharmacology, molecular cloning of drug targets. If you've ever wondered how scientists develop drugs that can effectively target diseases, you're in the right place. So, let's break it down together. First off, let's talk about drug targets. These are biological molecules, often proteins, that drugs bind to in order to elicit a pharmacological effect. They are crucial in modulating physiological responses in our bodies. In fact, about 40% of the action sites for most approved drugs are drug targets. Common examples include G-protein-coupled receptors, or GPCRs, and ligand-gated ion channels. These proteins act like molecular switches, helping to transmit signals within our cells. But why do we need to clone these drug targets? Well, cloning allows us to create multiple copies of a specific gene or protein of interest. This is essential for studying how these targets interact with drugs, testing the effects of potential new medications, and even engineering modified versions of these targets for research purposes. So, what exactly is cloning? In simple terms, cloning is the process of making an exact genetic copy of an organism or a specific DNA segment. Think of it like photocopying a document, but in this case, we're copying genetic material. A famous example of cloning is Dolly the sheep, the first mammal to be cloned from an adult somatic cell. Now, let's get into the nitty-gritty of how we clone a gene of interest. The process typically involves several key steps. First, we cut our cloning vector, which is a DNA molecule used to carry the gene we want to clone, using a restriction enzyme. This enzyme acts like molecular scissors, cutting the DNA at specific sequences. Next, we cut the DNA of our gene of interest with the same enzyme, ensuring that both pieces have compatible sticky ends. Then, we mix these two pieces together and ligate them, which means we join them using another enzyme called DNA ligase. Finally, we insert this ligated DNA into a host cell, often E. coli, where it can replicate and produce more copies. There are various techniques for molecular cloning, including restriction enzyme cloning, PCR cloning, and even more advanced methods like Gibson assembly. Each of these methods has its own advantages and is chosen based on the specific needs of the experiment. So, what can we do with molecular cloning? Well, it allows us to study gene function, produce important proteins like insulin, create DNA libraries, and even engineer mutant proteins to understand diseases better. It's a powerful tool in drug development and research. Let's talk about some common methods of molecular cloning. The first one is restriction enzyme cloning. This method uses special proteins called restriction enzymes to cut DNA at specific sequences. Imagine these enzymes as scissors that snip the DNA at precise locations, creating either blunt ends or sticky ends, which are essential for joining DNA fragments together. Sticky ends have overhangs that can easily bind to complementary sequences, while blunt ends are just straight cuts. This makes it easier to ligate, or join, the DNA fragments together using an enzyme called DNA ligase. Next up, we have PCR cloning. PCR stands for polymerase chain reaction, and it's a powerful technique used to amplify a specific DNA segment. It's like making a ton of copies of your favorite recipe so you can share it with everyone. In PCR, we use short pieces of DNA called primers that are complementary to the target sequence. These primers help the DNA polymerase enzyme to start copying the DNA. With each cycle of PCR, the amount of DNA doubles, leading to millions of copies in just a few hours. Another method is Gibson assembly. This technique allows for the joining of multiple DNA fragments in a single reaction. It uses overlapping sequences at the ends of the DNA fragments to promote the assembly process. Think of it as putting together a puzzle where the pieces fit together perfectly because they have matching edges. Then we have topoisomerase cloning, which utilizes topoisomerase enzymes to join DNA fragments. This method is super efficient and can save a lot of time compared to traditional ligation methods. Finally, let's not forget about recombination cloning. This method takes advantage of site-specific recombination, allowing for the transfer of DNA fragments between vectors. It's like swapping out parts of a car engine to make it run better. Before we dive into the tools, let's clarify some key terms you'll need to know. Drug target, these are biological molecules that drugs bind to, eliciting a pharmacological effect. Think of them as the locks, and drugs are the keys that fit into those locks. Vector, this is a carrier used to transfer the target DNA fragment into a host cell. It's like a delivery truck for our genetic material. Plasmid, a common type of vector. Plasmids are circular DNA molecules that can replicate independently within a cell. They often carry genes for antibiotic resistance, which helps us select for successful clones. Restriction enzymes, these are molecular scissors that cut DNA at specific sequences. They create either blunt ends or sticky ends, which are essential for ligation. Now, let's talk about the tools of the trade. First up, we have restriction enzymes. These enzymes are part of a bacterium's defense system against viruses. They recognize specific DNA sequences and cut them, leaving behind either blunt ends or sticky ends. For example, the enzyme BAMHI creates a 5-foot overhang, while KPNI produces a 3 overhang. These ends are crucial for joining DNA fragments together. Next, we have DNA ligase. After we cut our DNA with restriction enzymes, we need to join those fragments back together. DNA ligase does just that. It forms chemical bonds between the ends of DNA strands, effectively sealing the gaps and creating a continuous piece of DNA. Now let's discuss cloning vectors. These are essential for carrying our DNA of interest into host cells. There are various types of vectors, including plasmids, great for smaller DNA fragments, typically up to 10 kb. Bacteriophages, these can carry slightly larger fragments, up to 1 kb. Cosmids and BACs, these are used for even larger DNA segments, with BACs handling up to 300 kb. Each vector has unique features, like an origin of replication and selectable markers, which help us identify successfully transformed cells. Speaking of transformation, let's talk about competent cells. These are specially prepared bacterial cells, like E. coli, that can take up foreign DNA. There are two main methods to make cells competent: heat shock and electroporation. Once the DNA is inside, we can select for cells that have successfully taken up our plasmid using antibiotic resistance markers. Another vital tool is the polymerase chain reaction, or PCR. This technique allows us to make millions of copies of a specific DNA sequence. By using primers, short sequences that flank our target, we can amplify our gene of interest, making it easier to work with. Lastly, we have gel electrophoresis. This technique helps us visualize our DNA fragments. By applying an electric current to a gel, DNA fragments move based on their size, allowing us to see if our cloning worked as expected. So there you have it. These are the essential tools used in molecular cloning. Understanding these tools is crucial for anyone interested in molecular biology, pharmacology, or genetic engineering. In summary, molecular cloning of drug targets is a vital process in pharmacology that helps us understand how drugs interact with their targets, paving the way for new therapies and treatments. If you found this video helpful, don't forget to like, subscribe, and hit that notification bell for more science content. Thanks for watching, and we'll see you next time.
PharmaPal.